Hi. So Ms. Lalone was kind enough to run your Scantrons through so that you could get a grade and you could start test corrections. We feel it's really important that you do your corrections right away so that you're able to kind of move on to the next unit with a clean slate. So you've got 20 minutes, the clock's starting now. Ms. Woodall, if you don't mind passing out the Scantrons and the tests as I remind them of how to do test corrections. Remember that you do have to have a reflection. It needs to be at least half of a page. If you don't have that when you turn in your corrections, I am going to hand that back to you and ask you to do that because it's really important metacognitively. When you identify a question that you have missed, you'll pull out a word or phrase from it. You do need to record why you missed that question. Did you not study that topic? Was there something that really just confused you about it? Was there academic vocabulary you didn't understand? Was there scientific vocabulary that you didn't understand? So you're sorting or classifying the reason that you missed questions. And then you're trying to relearn that material. So two things about that topic. They can be two written things. They can be two pictures. They can be one of one and one of the other. But there do need to be two clear things. And they need to be fairly detailed. So for example, if your topic is the endomembrane system, and you just say it's a set of organelles in the cell, that's not in detail enough. That doesn't help you actually relearn anything. So you do need to be really detailed or I'll be giving it back, asking you to give it another try. So you have 20 minutes, starting now. Pause the video, work on your test corrections. When you're done, please make sure that you turn in the Scantron and the test. Of course, you've gotten the same test form that you took on Friday. Turn them back into the front. If you don't finish in 20 minutes, that's okay. You're welcome to finish sometime next week when I'm back, and you can work on it during lunch or after school. But we can only have 20 minutes because we have our lab to do today as well. So pause this and work on your corrections. Next, we're moving to start our inquiry lab. And so we kind of want to ask some questions about respiration and what is important to understand about respiration. So you're doing this inquiry lab and you're going to use a living organism, yeast. Remember that your package of yeast needs to be placed half in one flask and half in the other flask. So you only get one package of yeast. After you put half of the yeast in one and half of the yeast in the other, you need to add liquid A to the 100 milliliter line of the one flask. And then, to the second flask, you're going to add a known amount of sugar. Now that's up to you. There are scales on the cart. You can measure how much sugar you're going to add. And then add 100 milliliters or water to the 100 milliliter line. So now you have two flasks, one of mystery liquid A and one of water and sugar. You've added that to the yeast. I didn't say it here, but you do need to swirl them. You do need to mix them a little bit. And then you want to cover both flask tops with the balloon. And we talked about how to kind of hold it carefully so that the flask doesn't fall. Your balloon is kind of sitting down now. You're going to place both flasks on the hot plate with the setting no higher than level 3. Begin a timer. Record the time when the balloon stands up. Record any other observations. And then remove the balloon. Dump the contents in the sink. Rinse the flask. Rinse the sink. Put the balloon in the trash and the flask back on the cart, please. You do need to record all of this on your own piece of paper. These are a class set of directions. And then you're going to begin to work on three things. One, how did this experiment demonstrate the properties of respiration? How does baking bread demonstrate the properties of respiration? And I gave you a bread recipe. And then design another iteration of this experiment. Now we do only want to take about 20-25 minutes to do the lab. And so if you're not able to get all the way through these, that's okay. We can work on these again later this week. So if you'll go to your lab groups and go ahead and get started on the inquiry lab. But one more thing is that as you are working on the inquiry lab, you might have some downtime. And so I'm going to ask Mrs. Woodall to pass out to you this half sheet called a respiration pre-assessment. So while you're waiting for the yeast to perform respiration, you're going to kind of answer these questions in your group. And you can write on this. What is the purpose of respiration? Most people, when they give me their first thing that comes to mind, can usually be more specific later. And then you're kind of going through and answering these questions. So feel free to work on the pre-assessment while you're waiting for your inquiry lab to work. There is one other thing that we're going to be working on today, and that's your word wall. So make sure that you're not spending more than about 30 minutes on this section. Pause this video and then come back to it when you're done with your experiments. So you've done your experiment. You've seen that yeast, when given sugar, are going to produce some sort of product that you captured here in your balloon. But what product is it? The only way you know what this gas is by knowing that yeast is A, a living organism, and B, was given some sort of sugar. And so in this example, the yeast were given sugar in the water or sugar in soda. Now I let the soda go flat, so there wasn't any other gas in it. And so interesting to see how much sugar did you weigh and how much sugar was in 100 milliliters of soda. But because we gave the yeast glucose, they performed cellular respiration. Their point in performing cellular respiration was to make energy for their own cells. As a waste product, they produced carbon dioxide and water. So you watched this video last night. We're looking back at your notes to understand what was actually going on in that flask. There were yeast, there was sugar, there was water, there was warmth, and all of a sudden this balloon filled up. So what caused the balloon to fill up? So we should be able to look at the molecular level. Now remember we said that your yeast were probably not using oxygen because we didn't give them any extra oxygen. Putting the balloon on there meant no oxygen was getting into the flask. So they weren't using oxygen, but they were producing carbon dioxide gas. And so this, remember, is the overall general idea of what's happening. But we want to take a few minutes and remind ourselves that it's a lot more complicated than this. It doesn't happen all in one step. And the reason this is possible is because of redox reactions. So at the end of class today,
Mikhail Blake, meet your right out front immediately. Mikhail Blake, please meet your right out front.
You're going to work on your word wall. And working on your word wall, you're going to focus on this term redox. Use this picture down here at the bottom to help you if you want. Remember, redox stands for oxidized or reduced. When one thing is oxidized, it loses electrons. Where does that electron go? It goes to another molecule or element. And that molecule that gained electrons is called reduced. So we're taking some time to think about if energy is really in the bonds of the C6H12O6, that's where the energy is. And we're pulling that energy out via electrons. These redox reactions are what allow the cell to harvest and reuse some of that energy. The last thing that we want to keep in mind is that the electrons are actually going to be carried to do work of their own. So it's not that the electrons are given to ATP. That's what a lot of people think. They think that the electrons are taken from glucose and the electrons are given to ATP. And that's incorrect. The electrons are taken from glucose. The energy of those electrons is used to do work. And so that's, of course, what we're going to be exploring in this unit, is how do we get electrons to do work? For now, we want to make sure that we understand what a redox reaction is and what it has to do with cellular respiration. So, with the remainder of your class time, if you'll go to your groups, make your word wall 7 minutes to put the words down, switch 7 minutes to do connections, talking about redox reactions. Anytime that you have left, you want to make sure that you have finished the inquiry lab, finished the pre-assessment, and gone over your notes, and possibly finished your test corrections as well. today